Okay, here, the final chunk of this physiology of breathing chapter, also the final chunk of material before the third exam. We're going to look at, like I said in the last video, really for me, the pinnacle, the perfected lung, beautiful in its form and function. I cannot say enough things about it. I really think it's cool. And that's the avian lung. And the avian lung is going to be unique amongst lungs for us here because it is, my pen here, not a tidal lung, it is unidirectional. And we haven't talked about this because it's hard to imagine from the pictures that we saw. But even in things like alligators, right, which have that pump, right, that's moving air in and out of the lung, even things like alligators have a unidirectional lung, right? It's true of, as far as we know, all of what we call archosaurs, or ruling reptiles. So that's alligators, crocodiles, gharials, caimans, dinosaurs, okay, and including birds, at least most dinosaurs. Now, I mean, some dinosaurs may or may not have had it. It's another story altogether. But only in birds do we see this really beautiful, elaborate expansion of this respiratory system into all of these wonderful little parts. And excuse me, like I said, this is like the 9th video I've done today, and I'm going to be unable to speak after this probably. But why this is so important, right, is that with the tidal lung, we're still stuck with stale air, right? And if we're going to be a bird and we're going to be flying, right, we can't really work with stale air. We have to have as much fresh air as we possibly can in order to really make that energy expenditure possible. And so what we end up with is a lung, right, here in the middle, and a series of these accessory sacs all around it, right? So there's, and the number of sacs varies depending on the animal. Here we've got a goose, right, with a cervical sac, and an interclavicular sac, and a couple of thoracic sacs, and an abdominal sac. And this is a pretty typical arrangement here. And these are not for gas exchange. These are simply air storage facilities. So if you remember back to how we used oxygen in the oxygen transport chain, right? We passed electrons, or electron transport chain. We passed electrons from hand to hand to hand to hand to hand to hand, right? These sacs are going to have air in them from one to the next to the next to the next to the next. And so we're going to see this continual flow from one sac to the next to the next, eventually through the lung and back to another sac and then back out. Okay, so they do still have a trachea, like all normal things do, right? Instead of having a larynx, they have a structure called a syrinx, right? If you're familiar with Rush 2112, they're like their big famous song, Temple of Syrinx, right? The syrinx is the sound producing organ in a bird. It's also where the glottis resides. But air passes from the trachea, across this syrinx, and then into these structures. How this works, and we've got a little video, and we've got some animations and stuff to try to get this around you the idea here. But we have a complete separation of inhaled and stale air, right? So during inhalation, what happens is these posterior sacs expand, okay? And as they expand, they suck air in, fresh air in. So it passes all the way back here and it fills this posterior sac. The anterior sacs also expand. And as they fill, they pull air from back here forward. And as they pull air forward, they come across the lung. And it passes through these structures here called parabronchi. And this is where gas exchange actually happens. Okay, so we're sucking fresh air in back here, and we're filling this space. We're sucking stale air from here forward, and as we're doing so, we're exchanging gases. Then when we breathe out, both of these sacs contract. Stale air gets forced out, fresh air gets forced up and into this space and across these parabronchi again. So let's see if we can watch that in action. I have no idea if this is going to work. Animations have not worked very well so far, but we'll see. At least hopefully the video will work for us. No, obviously this is pretty cartoony, but so these would be like the interclavicular air sacs. These, of course, would be the abdominal air sacs back here. And there's no sound. Well, we're following one inhaled breath all the way through. Okay, and there's our second one coming in, and you can see they are not mixing. So we are exposing our respiratory membrane entirely to atmospheric air. There is no stale air there. And as we do this, right, we can further enhance this by having blood flow cross, either as countercurrent exchange, right? Sometimes it's cross current exchange. But again, doing the maximum, right, in terms of picking up oxygen at those parabronchi. And again, we could watch this all day. I'm not going to ask you to watch it all day, but that's more or less how it works. Again, I find it fascinating, absolutely beautiful. So here are those parabronchi. They are different. They're not really cameral. They're not really uni or para or any of these cameral. They're just kind of fascinating in their own beautiful little way. In the world of invertebrates, right, so outside of things with backbones, we rely on a number of different possibilities here. For aquatic things, we have gills in a lot of cases, although not all cases. So we talked about horseshoe crabs before and their gills and their relatively interesting respiratory cleaning cycle. They have what are called book gills, right? Because their gills are big flat sheets that are arranged like the pages of a book, right? We've got things like polychaete worms that have this sort of fan of respiratory tissues on these little tentacles, called a tentacular fan. Some polychaetes have little gill tufts instead, right? We get weird things like echinoderms that have a water vascular system. Okay, so they have kind of a conjoined respiratory vascular system where they pull in water through their tube feet, and that water then serves as their blood supply, basically. And they've got on the dorsal surface, right, all of these little papillae, right, which are also for exchange of water, both in terms of the fluid within and also exchange with the respiratory system. Very complicated, very unique. If you can remember water vascular, I'm happy with that. And then we get into things like gastropods, right? So aquatic gastropods, things like snails, clams, again, have gills, right? These gills reside within a cavity called the mantle, right? And that mantle in both a snail and a clam is within the shell, right? Ventilation of these gills occurs different ways. For an aquatic snail, they can pump air in and out of this mantle cavity. It comes in one side, passes the gills, and goes out the other. For things like clams and other bivalves, they open and close their shells, right, to suck in and push out, suck in and push out. Some of them have siphons, okay, so they can keep the gill or keep the shell closed essentially all the time. They can open it, they can move it a little bit without opening it and suck air in or water in and out through these siphons, an inhalant and an exhalant siphon accomplish the same thing. So things like gooey ducks, other clams that bury themselves, that don't necessarily want to expose everything to the outside world, can use these siphons instead. For shellless gastropods, for shellless mollusks, we get things like squid. They're not gastropods, they're mollusks. We still have a mantle cavity, right, as before, only now it's not covered by a shell, it's just a bit of soft tissue. But they use, very much like our gooey duck here, siphons. By expanding and contracting this mantle cavity, they can suck water in, ventilate the gills, and force air back out, or water out rather, through a exhalant funnel. A lot of these animals use this funnel for jet propulsion as their primary means of moving around. Land snails are called pulmonate snails because they don't have gills. They have a lung cavity. They don't really have a lung, but they have a lung cavity. And the exchange of gases occurs through the skin here within this lung cavity. They ventilate it the same way that our aquatic snails do here by moving their body up and down within there. For arthropods, we get a little bit weird in some cases. They have this fun little suction thing that they do. So their gills are underneath this plate behind their head called the carapace. So they're protected within the carapace here. And there's a little opening at the bottom where their legs come out. And at the very front of this chamber, we have a little plate here called a scaphognathite. And this scaphognathite flaps up and down. And as it flaps up and down, it sucks water into this chamber, right, the branchial chamber, right? And as it does so, right, it sucks water in and pushes it out, sucks water in and pushes it out. And in doing so, it ventilates these gills. For arthropods on air, or that are air breathing, obviously we can't do that. And so there's a couple of different means by which we do that. You can actually see, well, maybe you can't see, but there are little pores back here along the abdomen. These are going to be the entrances and exits of the respiratory system in a lot of our arthropods. Some arthropods are going to have, like spiders, for example, are going to have a chamber back here called an atrium, where their respiratory membranes reside like the pages of a book. This is a book lung, not like a book gill, but a book lung. And there's a little opening here called a spiracle. Now, for these guys, sometimes they can actively ventilate a book lung, right? They can move the outer part of this carapace up and down sometimes, not always, but sometimes a little bit, and actively ventilate it. But for the most part, when we talk about insects, all of the ventilation of their bodies happens entirely by passive diffusion from the atmospheric air out here and the atrial air in here. And as you might imagine, that's going to severely limit their ability to bring in oxygen, right? One of the reasons we don't see giant insects anymore, there are a variety of reasons for this, but one of the reasons we don't see them is because there's not enough atmospheric oxygen anymore for bugs to get above a certain size. Back in the Triassic and Jurassic and Cretaceous, the air was a lot more oxygen rich than it is today, right? And so there was more oxygen. Diffusion worked better in those cases. They were able to get more oxygen per unit time. They got big, they got massive, but they simply can't today because of this limitation. Outside of the realm of the spiders, okay, most insects, again, rely on these spiracles to be their entryway, their entry point for gases. Within their bodies, however, these spiracles are attached to these little tiny little pathways here called tracheae. Right, so not like a trachea, like you've got a singular one, but a lot of separate little pathways, the tracheae. You can see a little x-ray of a beetle, and these little white lines in here, these are all those little tracheae, and they're each connected to a little spiracle. Now again, right, entirely on diffusion, right? There's really very little in the way of active breathing. It's almost entirely passive. So air exchanges across those spiracles with the air in the tracheae and brings gas into the body that way. It's not very efficient. right? It's really not. The oxygen cascade here is pretty intense, right? So again, we're really limiting the ability of insects to really have activity at a very high level, right? And certainly at a certain size, they're just incapable of getting past there. Okay, so this concludes this particular unit. Well, everything up to this point will be on the third exam, and everything after this point will be on the final exam, which is, of course, remember, not cumulative. So study this stuff, take the third exam, and then we'll worry about the stuff after that.